not final, okay, until I say it's final. So don't worry about it. We'll fix it. Just it takes some time, okay? Don't be mad. I'll go over if there is serious problem. But it's some sort of guiding you how to study for the exam 3, this quiz, okay? You will have to say about this mechanism. For instance, how HIF1 is regulated by oxygen. You just can say the high oxygen HIF1 gone and low oxygen. No, okay? We really have to say about BHL, okay? PhD. Even if you don't remember the protein name, you'll be fine. But you really have to understand, okay, how it's regulated. Like it's modified ELIS or hydroxylated kind of stuff. Okay. All right. I'll talk about molecular targeted therapy. Now I kind of understand why we need these types of treatment. Because of heterogeneity, the chemotherapy is very non-selective, although it's relatively selective because it's targeting highly proliferative cells. But I'll talk about this, and I'll talk about immunotherapy later. And also I incorporate a little bit of transgenic animal model because I really want to introduce some updated region studies, but without talking about this transgenic cysta, it's very hard for me to explain. So I'll spend a little bit of time. It might be a little bit too much, but I think TA can help you out. All the TAs, except undergraduate, already took my graduate course, so they know. So again, this is the future. It's kind of, we call it the personalized medicine or precision medicine. Is not only on top of the conventional diagnosis, we're going into very deep at the molecular level, identify what kind of mutation this individual patient had, and then we design a treatment according to that mutation. So for a long time, there is some clinical trials. So you have some very nice chemical identified through some in vitro research or in animal research like we're doing in my lab. And we have some pre-convinced evidence in my work. And we go to clinical trial, we collect the patient and do like very expensive clinical study, just looking at toxicity, all different things. And sometimes this chemical gave us like only 10% of patients are responding, okay? And previously, the 10% wasn't enough, and they just dropped the drug. Now we know how stupid we are, okay? The 10% could be very nicely responding to that, whatever the drug we're testing, okay? Think about it, heterogeneity. Every patient's tumor or cancers are different. So we are really looking at the very specific population, even if it's just 2%, 5%, doesn't matter. And even the insurance company can acknowledge this, so they kind of try to cover, even for those small populations. It's all about money, because it's not that big market, then kind of hard to develop, but that's what we're trying to do, right? So it's very different in terms of how we interpret those big studies like clinical trial. So I'll just briefly touch again. Chemotherapy, just want to emphasize how it works. Mechanism, okay? I want you to understand the various mechanisms, but bottom line is targeting highly proliferative or rapidly dividing cell, which means it's not selective, it's not targeted. It's a very kind of very crude way, just there's no targeted. So again, this is why it's not really less than 50% in most cases. Cancer cells are not that highly proliferative, so that they are not really, they don't really care about this chemotherapy to survive. So again, we talk about this EGFR receptor. So I don't really have have to go over again, but again, it's all about control or regulation. We really want to amplify, proliferate, or make it double our number of cells or grow when only we need. So it's all about control. Otherwise, it's very bad. So these growth factors, ligands, hormone can bind to receptor, and there's activation cascades, usually through the phosphorylations. So this is basically EGFR, same family members as her. This naming is kind of very messy because based on who and when discover or identify this gene, they just name it. But the herd is basically human EGFR. It's basically EGFR family member. There are many family members. So herd 1 is EGFR, that we talk about this, okay? And there's herd 2 is EGFR2. So there's many family members, similar but a little bit of different in terms of structures and function and tissue distributions, all different things. But basically this HER2 has been known quite some time that this HER2 is amplified, okay? This is not mutated here in this case, although there is a mutation, there are some, but it's mainly all amplified. So the gene of this HER2, that region of chromosome has amplified because of this defect in DNA repair system. Okay, so we have some sort of problem in that repair system that somehow is amplified. So it's over 25% of breast cancer. HER2 is amplified, so we call it HER2 positive, and it's overexpressed because you have a multiple gene, more than 2 genes, so that you end up higher levels of HER2, mRNA, and protein. Okay, because again, the HER2 is EGFR. This is signaling pathway. It activates or promote proliferation, survival, and suppress apoptosis. Is all these really bad thing in cancer. So if you look at the survival rate, if the patient's tumor breast cancer is HER2 positive, then median survival is for 3 years. But if it is HER2 negative, if there's no amplification, then it's doing a little bit better. And also many other types of cancer. I just delete all this because it's too much. But lung cancer and many other cancer is overexpressed. So people got very interested in what about targeting this. So actually this is, I'll tell you, this chromosome, and we kind of make some probe very specific to this HER2 gene. So I think I label as green. Okay, so it makes sense. Only 2 copies, right? But this is HER2 amplification tumor. It has a lot because the gene area is amplified. Then this is so then this is HER2 positive tumor, and this is a normal tumor. Okay, when you have some suspicious nodule in your breast, then you do some biopsy and send it to histology or pathology. They do staining called immunocytochemistry, and you see that if it's positive by amplification, then you have more HER2 expression in the tumor. Then it came out, okay, it's HER2 positive. All right. Then this again, this is very important. HER2 is basically it all doing a survival and proliferation. Then this is kind of I just found this very nice some sort of cartoon or animation. So HER2 and sometimes it's HER2 homodimer, but they form with the heterodimer with other family member. But that's not important. It's activate this cascade. Signaling cascade, decrease apoptosis, increase survival, increase cell cycle and proliferation, and also increase angiogenesis. It's all about known to promote cancer progression. So this is some sort of a first wave of molecular targeted therapy. So this is not some sort of chemical, it's antibody. Okay, it's antibodies specifically recognize and neutralize HER2. So I think you've heard this is a very famous drug, Herceptin. And it's very expensive still. I think this is outdated, but still, this 1 year treatment it costs about between $70 to $100,000 if you don't have insurance. And insurance probably a little bit better, but still, it's very expensive. And it basically it recognizes HER2 receptor and suppress this HER2 signaling pathway. So you see the difference. It's very different, okay? Instead of targeting highly proliferative cells, it just targets the HER2 specifically. So it's more susceptible or more effective when the tumor cells express or overexpress HER2, okay? But think about the tumor that doesn't express HER2. It's no use, right? There's no reason to use. So this is which is better. Also, HER2, if you because it's antibody, also it kind of triggers the immune system. So it's basically a very early stage of kind of phase of immunotherapy. But it's this is kind of concept for.
Herceptin, a multimodal approach targeting HER2-positive breast cancer.
Some sort of
For some women,
commercial.
effective treatment of breast cancer can start deep at the root of the disease. Some of the most aggressive breast cancers are driven by a transmembrane receptor protein known as HER2, a member of the HER family of receptor tyrosine kinases. Roughly 20,000 HER2 receptors are typically expressed on the surface of healthy breast cells. In approximately 25% of primary breast cancers, the HER2 protein is overexpressed, resulting in tumor cells with as many as 2 million receptors present on the surface of each cell. These cancers are called HER2 positive. The effect of HER2 overexpression is a tremendous increase in receptor-mediated intracellular signaling, directing the cell to proliferate uncontrollably. For this reason, HER2-driven disease results in an aggressive form of breast cancer, which, if left untreated, can progress rapidly. Herceptin is a monoclonal antibody specifically designed to target the HER2 receptor. With an extended half-life that allows for constant exposure to tumor cells, based on preclinical studies, Herceptin delivers continuous inhibition of HER2 activity by working on both the extracellular and intracellular domains of the receptor. Herceptin is the only approved therapy designed to bind to HER2-positive tumor cells extracellularly and flag them for destruction by the immune system. In addition, bound Herceptin prevents HER2 receptor activity by blocking intracellular HER2 signaling and ultimately inhibiting cell proliferation. Along with its direct effects on the HER2 receptor, based on preclinical studies, Herceptin can enhance the effects of chemotherapy, leading to cell stasis and death. With several distinct mechanisms, Herceptin works both extracellularly and intracellularly to deliver a multimodal attack against cells overexpressing the HER2 receptor, a receptor linked to one of the most aggressive forms of breast cancer.
So this is amplification. So still it's under control. Still it requires the ligand or growth factor, but it's highly expressed so that it's critical. But as you can imagine, this is a HER2 positive tumor if it's basically seeing this type of staining. And pathologists will diagnose this tumor as a HER2 positive. What do you see here? So this is HER2. You can make it brown, whatever, green, red. It depends on your preference. But not all cancer cells are HER2 positive. Again, we have the same problem, right? Like a proliferation. This is one big giant tumor here. I think it's a mouse tumor that I don't even remember where. But for some reason, remember clonal evolution or stem cell mutation, only half of them are HER2 positive clone, for instance, and this is totally negative. If you use Herceptin the Herceptin is not the only one that is kind of commercial and already very long time ago. There are many types of or different types of treatment available, and so basically we'll have the same problem. This HER2 positive cell is going to be very unhappy. Those Herceptin or this monoclonal antibody will kill all those cells, but these guys really don't care. Even worse because it's very selective to HER2. Okay, it's the same reason why it's heterogeneous. So and then we can move on. Yes.
Is that selectivity part of the reason why you target it with chemotherapy as well?
That's good. Yeah, I'll get to there. So basically, right now we try to combine with other chemotherapeutic agent and different types of molecular targets, just increase the target kind of repertoire. So yeah, it's a good question. And no one right now, there's a lot of study. I'll show you some slides, but there is monotherapy with Herceptin only. Usually we don't do that. We just always combine it with some other chemotherapeutic agent or radiations or other types of molecular therapeutic target. Yes, that's very good questions. So still is over-amplified, still like wild type under normal control, normal regulation. But this drug is not antibody, it's a small molecule, okay, inhibitor that specifically binds to this ATP binding site. Because this ATP is very critical, because that how they phosphorylate this downstream target. So we try to target that very key site. Okay, so this is another type of molecular therapeutic kind of drug, erlotinib. So this is EGFR inhibitor, basically targeting a similar, same family member, the HER2 or EGFR. The reason is because it's not about amplification. This EGFR, so it's EGFR1 or HER1, is the third most common mutated gene in lung cancer. It's a non-small cell lung cancer. So it's P53 is always number one, majority of time, and KRAS, this STK11, I'll tell you more. And EGFR, okay, or third or fourth, the most mutated. So we want to target, I remember this slide, right, these types of mutation that we found in a real human cancer. So by mutating that area, which is very key, the region that is ATP binding site or the phosphorylations, or even there's mutation or deletion in the upstream, in the extracellular. So you get rid of, if you just lose this outside, then somehow it just constantly active without binding to the ligand or growth factor. So whatever, there are many different types of mutations. Some mutation is very lethal, so that there won't be any protein, but some will acquire this constitutively activated or oncogenic capacity. Okay, we really want to target this mutated EGFR. So basically, again, I show you this, the EGFR mutation. It's very rare in this specific lung cancer, squamous cell, so you don't want to use, right, in squamous cancer. But even in adenocarcinoma, actually this drug, erlotinib or EGFR inhibitor is widely used in adenocarcinoma, but not all adenocarcinoma patients. This is categorized based on the histology, right? But we really have to look at the mutation, okay, among the adenocarcinoma patient. Even we just sent out this biopsy sample to pathology, and pathologists, okay, this is adenocarcinoma based on the morphology or staining pattern, but still we really have to do sequencing. So now they send that to the molecular diagnostic lab to the sequencing, and only 10 to 40%, if the Asian population is very high, still we don't know, and usually like between 20 to 30%. And if they came out positive for this EGFR mutation, we kind of treat this patient with EGFR inhibitor. So this is one example. With 8 weeks of erlotinib or EGFR, so it is EGFR mutant tumor. The patient has this EGFR mutation, and after this, it just works we choose, okay, you are a candidate for this targeting mutation. It's very different from chemotherapy application, right? We just try to mix a bunch of different types of combinations and see if it's responding or not. It's kind of very blindly. Now we know what we are targeting, the specific types of tumor, not just one tumor. That among those tumors that we try to identify that whether this tumor has a specific mutation in this specific oncogene or not. Okay. So after that, this is big. It's still kind of very, very active and widely used, although I think it's quite expensive. But there are many other types of chemicals or antibodies or inhibitors basically trying to target this EGFR mutant tumor. Okay, so there's multiple drug from multiple company that we have. So another kind of example. So it's ALK. This is a very active, important kinase. Like again, it's kind of kinase signaling pathway. So because of the genome is unstable through all this defect in DNA repair system, that this kind of rearrangement of this region in chromosome 2 is very frequent in lung cancer and many other cancers as well. So this ALK, this is kinase, somehow rearrange it and form kind of fusion protein with other types of protein in EML4. Okay, so we call it ALK EML4 ALK fusion. Still, I don't think that we still fully understand why this ALK, this kinase, become constitutively active when it fuses with AML. But the bottom line is it's constitutively active. So by fusing with this protein, it makes it very constitutive. And this is basically the kinase activate the similar pathway that the EGFR can activate. So it's very, very highly proliferative signals coming in. And again, by fusion of these two protein, it become, again, constitutively active, okay, with a lot of control. Again, if you look at this ALK fusion here, right? Again, squamous cell carcinoma is very rare. So that's why we're studying the squamous cell carcinoma, because there's no obvious target or mutation that we can target. But it's about the 7% to 10% of adenocarcinoma has this kind of fusion. Okay? Again, there's a multiple target potential candidate. We get the sample and do the sequencing, and they kind of have a profile and determine which targeted therapy we're going to use. Same. This is targeting the ALK. Again, from here, it's very very bad, but it's very effectively treated those patients. Okay. So this is molecular targeted therapy. Again, so now you understand we would have to know the genetic profile of the cancer. Okay. We're not anymore depend on the histological diagnosis or classification. We're going deeper. So that's why this next generation sequencing, deep sequencing, it kind of revolutionized all this clinical aspect, because now it's quite inexpensive, economically doable, and it will be cheaper and cheaper. So we can sequence or look at the genetic profile of individual tumors, all right? And then we determine the therapeutic, okay? But only 7% out of the many lung cancer patients will respond. Still, it's very important, okay? Because for a long time, we think the cancers are same, homogeneous. That's why it probably, like 20 years ago, 30 years ago, this will not kind of be pursued and FDA approved, because it's only a small population. But now we know this, not every single tumors are different, so we would have to target specifically, like we call it personalized medicine or precision medicine. Okay? So here it stands, key point. There is no molecular targeted therapy developed so far. There are two, I think, I remember, past few years, still not very effective, but there are way more options here. this is mainly, not mainly, but it's female non-smoker has usually have this adenocarcinoma. It's famous, it's just basically smoking. But this is a kind of like related to the question that the original studies, very original studies. So I mean, the numbers are kind of outdated, so it's not accurate. So just don't care, don't pay too much attention to number. But this Herceptin showed that it improved overall survival in the late stage, with the late stage, only about 5 months. So it's not cure, okay? I mean, many regions, but see those heterogeneity, okay? The tumor will regrow. So basically, the resistance to the treatment developed rapidly in virtually all patients. The same problem, okay? So that's why we really want to combine with other therapeutics. So at least the breast cancer we did a really good job. The survival rate that can kind of steadily increase. So all these types of study and still lung cancer and pancreatic cancer is really doing bad. So what about then? It's not about just tumor intrinsic mutation. We talk about tumor microenvironment, okay, especially in the pancreatic cancer. Again, I showed this many times because I think it's important. This fibroblast is doing a really, really important role here because there's lots of fibroblasts there. But it's not just there. They're interacting, they're affecting, they're influenced by cancer cell, and then these activated or educated fibroblasts or cancer-associated fibroblasts that are talking back to cancer cells or other cell type. So I want to introduce this very exciting study, although very confusing study. So this tumor-associated fibroblasts, okay, like macrophages, this is one of many examples. This is a cancer cell. I think it's a colorectal cancer cell, so that's not important. But if you just inject these cancer cells alone into these nude mice, the tumor is not growing well. But if you combine, so you just prepare the cancer cell and also cancer-associated fibroblasts from real tumor patient, you mixed it and inject it, and then now your tumor can grow. Okay, these cancer cells are same, it's no different. But only when combined with fibroblasts, tumor now not just increase, this is kind of very different types of cancer, right? But only difference is without fibroblasts. Fibroblasts is not cancer cell, okay? They are not proliferating, they're just there and doing something to this cancer, okay? Because of the cancers grow so fast so that they lose body weight. But this is just cancer cell only, which is not very tumorigenic in nude mice. But when you combine with cat, so huge tumors, it's a very dramatic effect, okay? We're not manipulating anything in the cancer cells, only adding fibroblasts. Now you have a huge increase of tumor growth. So there's many other studies there. So now we kind of thought that this fibroblasts, by interpreting this data, everybody agreed that tumor-associated fibroblasts promote or inhibit cancer. No? Promote, right? It accelerates tumor progression, not inhibiting. So that's what we thought. That's kind of what we believe, that tumor-associated fibroblasts or cancer-associated fibroblasts promote tumor growth. So this study just came out probably, when did it came out? Not here, a couple of years ago. So they really want to kind of test in vivo, just in a more clinically or biologically relevant setting. So they kind of abulate, they kind of get rid of all this fibroblast population in the cancer, okay, and see what happens. So if the cancer-associated fibroblasts promote the cancer progression, and if you get rid of a cancer-associated fibroblasts, then what do you expect? The tumor growth will decrease, right? That's what, and they found it the complete opposite. It accelerated pancreatic cancer, and the survival rate is very dropped. Okay, so why? Before that, I really have to talk about the animal model. So even if it's kind of a little bit complicated, just follow me, okay? I'll go back to HIF1, okay? Don't worry about HIF1 is not important here. because I just have a slide, so I don't have enough time to make a new slide. So if you want to study the function of this gene, HIF1, then the one way of doing it in very direct way is we get rid of this HIF1. See what happens, okay? Then we just analyze the phenotype. Without the HIF1, what happened? And then we okay, the HIF1 is important, what, okay? So here, that's what we did like long time ago. Not I did, but actually my postdoc mentor did. So you get rid of the whole body. You don't worry about how we make this whole body knockout or transgenic line. It's not important. Just we somehow we get rid of this HIF1. So every single cell in this mouse has no HIF1 at all. And this is embryonic development. If you can say it's wild type, if you get rid of it, it's kind of abnormal already in 8 days. And then in between these 8 and 9 days, this embryo died. Okay? That indicate what? HIF1 is essential gene for proper embryogenesis and survival. That's all we can know from this study. Right? It's a critical gene, essential, this important information, but how and why? But because these embryos die, we cannot study because there's nothing to study. We're not going to get any pups because it dies during embryogenesis. If it's a very important gene for our whatever the biological process, survival, and if we get rid of it, gone. We're not going to get any of these mice. Or we can analyze this embryo a little bit, but it has very, very limited information. So basically, vasculature is one of the mechanisms why the HIF1 is so important. This is important for, because if you knock out HIF1, you don't see proper vasculature. So then if you don't have a vasculature, then probably you're not going to get any oxygen, nutrient, or many other stuff. So that's one of many potential mechanisms why the HIF1 is so important. Okay? So HIF1 is basically is important for hypoxia, right, and low oxygen tension. So that HIF1 activated on low oxygen tension, and one of the mechanisms of HIF1 increase angiogenesis, right? So this is part of the region. So, but we still want to study HIF1, okay? So that's why we go into this Cre-Lox-P site. I think TA explained a little bit, but I want to go over one more time. I just want to help you to understand. I want you to understand this, okay? So this is totally transgenic, artificial. This is not present in our body, in mammalian cells. This enzyme, this Cre, is a recombinase. It's from bacteriophage, okay? So we analyze this, some sort of virus, and take this gene out, it's a recombinase. And what this Cre recombinase does is they catalyze the site-specific recombination. So they just recognize this very specific 34 base pair LOX-P. We call the LOX-P site. Okay, this Cre enzyme recognizes this site. So if we engineered whatever the gene, so this is HIF1, for instance, HIF1. Imagine that this is HIF1. We put this LOX-P site flank the HIF1. Okay, we can do all this engineering. Don't worry about the detail. Then we add the Cre by many different approaches. Then what happened is it recognized this specific LOX-P site and do this combination, and then the result is this delete the HIF1, whatever the gene, the flank in between the LOX-P will be gone. Okay, this is how we get rid of whatever gene you're interested in. All right. So this is how we make this. Then we go back. I just put the video. I will not play. I want you to watch later. But I just want to refresh because I realize that not many of you really don't fully understand this transcription machinery yet. So again, there is the tissue-specific expression. For instance, skin cells or keratinocytes express a certain gene, certain protein that you don't see in a hepatocyte or neurons, right? But we have all the same genomic, all the cells have the same genetic information. we have a very different types of gene expression profile. And mainly it regulated by this transcription. So we have this transcription, this gene of interest, whatever we are looking at, and there's promoter, and the specific cell type or specific tissue has its own transcriptional program or transcriptional complex. So like skin cell has different kind of a transcription factor that only active in the skin cell, but not in hepatocyte. That's how we see specific cell type specific or tissue specific different program, right? Because skin cell and hepatocytes are very different, right? They don't have the same whatever biological process or gene expression profile. They're very different. So we depend on this promoter specific activation. So again, we're going back. So we put this engineer the specific promoter. So here is again is K14. Now we know that keratin 14, this is a very important gene you make for the keratin in our skin. So it's only expressed in skin cell, not in hepatocyte or other cell type, because it's very specialized protein. Keratin 14 is only expressed in epidermal keratinocyte. So that this means that keratin 14, there's a gene, but in front of it there's a promoter. Okay? That's only active. That promoter is only active in skin cells, right? Determined by specific transcription machinery, different types of transcription factor, right? You understand? You will have to follow this, okay? So now we all know the sequence, so we take this promoter region of this gene, not the gene, the promoter of K14 gene, and in front of the CRE, just basically we'll just copy and paste, cut and paste into the mouse genome. So this mouse usually don't have this K14, the CRE, because it's from where? Bacteriophage, it's totally artificial gene. We engineer to have this CRE gene into the mouse, but not just this CRE, we just add in front of the CRE, we put this specific promoter. For instance, this K14 promoter, okay? So that then this CRE is only expressed where? Or this promoter become activated only where? Skin cell, right? Only those skin has a specific transcription machinery to activate this promoter so that you have CRE. Although this transgene is in every single cell in this mouse, right? But only skin cells can react to this skin kind of transcription profile or machinery only because of the promoter, and then you activate it. You understand? So CRE is only expressed K14, and now we have a target. So it could be HIF1, I want to study, but the other way around. If in addition to knocking out or deletion HIF1, we can make it constitutively active. How would you do it? Remember how HIF1 is regulated.
Knockout VHL.
So VHL is very important because if HIF is modified, then VHL recognize this modified HIF1 and target for proteosomal degradation. What do you think is going to happen if we delete VHL here? Yeah, HIF1 will be stabilized even in the presence of oxygen. The cell kind of confused that even in the presence of oxygen, still they think that, oh, it's hypoxia because the VHL cannot do their job because there is no VHL. So we flux, again, put the LOX beside and the flag is VHL gene. So we combine this, we breed these mice, and then it's the same cells express CRE and also have this VHL. But this VHL will be deleted only what cell type? Skin cells, okay? Again, if we delete this VHL whole body, there's no mouse. There is embryonic insult. It's important. So we want to study this. Again, you can do the same thing if you want to get rid of a HIF1 only in skin cell. That's what we did actually. Then this is how we get. So just try to understand this transcription, okay, the promoter specific, so that we can do conditional. This is basically spatial control of expression. We don't want to get rid of whatever gene you're interested in, the whole body, the specific gene we want to study. Then this mouse may be survived, may be viable, right? Because it's still other types of. So again, like a VHL is gone only in the skin, and if you don't remember the HIF1, the VHL is gone. Then still, even in the presence of oxygen, this HIF1 will not degrade. Okay, here the low oxygen, but because of the VHL is not present here, still in the normal oxygenation, the HIF1 is activated. So we can study the HIF1. Okay, so it's basically overactivated HIF1 and analyze the phenotype. Kind of we can interpret what the function of HIF1. And among many target genes, because it's a transcription factor, this is the VEGF, angiogenesis. There's many other factors. There's VEGF. So think about it. The skin cells express tons of VEGF all the time because they usually need this VEGF when oxygen level drops, right? That's kind of homeostasis. But when we get rid of the VHL, then the VHL is constitutively overactivated like cancer cell, so that we have more blood vessel, and this is why we have more blood vessel in the skin. Okay, so this is the K14, same gene, same skin, and the VHL is gone, so that because you have a more and dilated blood vessel, so you lose very red and lose heat very rapidly because the subcutaneous, our cutaneous vasculature is very important for heat or temperature control, this kind of part of it. So it's not important this specific detail, but I want you to understand how we make a specific knockout, conditional knockout we call. But we want to do more. In addition to this location or tissue specific type, we really want to control the timing as well. Okay, we want to delete or activate or anything, manipulate when we want. Okay, so I want to study some aging-related gene. Okay, so I want to wait until the mice become very old, and I want to get rid of this gene. So you have to wait until. So this is one way. So remember the nuclear receptor pathway. What is the nuclear receptor? All right. So nuclear receptor is a transcription factor, okay? But it usually stay in the cytoplasm. The transcription factor should be in the nucleus, right? So that they kind of bind to DNA and activate the transcription. So when they in here and there is a signal, ligand, for instance, but usually in this case is steroid, the hormone. And hormone, especially steroid hormone or testosterone or estrogen, that because that is a lipid soluble, it can actually pass the membrane. And if this hormone or whatever ligand binds to this nuclear receptor, then it can go to the nucleus. That's how we control this transcription, the nuclear receptor signaling. So only when the hormone is there, the receptor is kind of can go into the nucleus and activate the transcription. So we can control his. So in addition to this promoter, CRE, we add the estrogen receptor, any types of receptor, but estrogen receptor works pretty well. Here, so because of this receptor, estrogen receptor, nuclear receptor, it will stay in the cytoplasm, okay? But only when we add this tamoxifen, which is similar to the estrogen, now it binds to this nuclear receptor or estrogen receptor. Now this whole complex, fusion protein, go into the nucleus because CRE needs to be in the nucleus. Why? Because that's where the DNA there. You understand? So even if you have this gene, it's not gonna be in the nucleus, so that there is no cleavage or no modification. Only when you add this ligand, we can add the ligand, we can inject it or add this in the cell, then this it binds, and the nuclear receptor can go into nucleus. Are you following? You will have to understand this, otherwise you're very unhappy in the just read. Okay? So when we have this, then you can control when you. So you just wait until, or you can study when the mice are like 6 weeks old, or 6 months old, or 2 years old. Okay, you can just inject this tamoxifen or any types of chemical so that you activate the nuclear receptor. Now this whole complex can go into nucleus and cleave whatever you want to cleave. Yes?
So to be clear, this receptor create complex is expressed throughout life, but it's not activated until you add this tamoxifen.
Yes, it's expressed whole and every single cell type. But only when exposed to this ligand and it can go into nucleus. Again, this Cre, because of this promoter, only expressed in certain types of tissue, right? So we have a spatial control and also temporal control. So this, because of the promoter, whatever promoter you put in, is only expressed in the certain types of specific types. So for instance, if you have a K14 promoter Cre, and this only expressed in where? Skin. But this guy not doing anything because they are in cytoplasm. Okay, if they're in cytoplasm, how can a Cre do their job? Because they have to go into DNA in the nucleus. Only when exposed to this chemical, and like this typical is the mechanism how this nuclear receptors work. Only when bound, it can go into nucleus and then do their job. You understand? So we can control the location of this Cre activity and the timing. Okay? Got it? That's how we control. Alright, so you remember this video, right? So now you should understand how we make this video. So okay, it's not about just deleting or anything. You can do whatever you want to do. It's basically engineering our genome. You can do whatever, as long as whatever you can test it. So here we put this GLP, this green fluorescent protein, right, from the jellyfish. And this again is artificial gene. We don't have GLP. We're not green under under UV, right? We make them green if you want. But we put this stop kind of a sequence in front of the promoter. So this, because of this stop, this transcription, and kind of hold there, it's not going to be complete. So the GLP will not be expressed. Okay? But we put this LOXP site between the flank to the stop. Think about it. There's a LOXP here, stop, LOXP. Okay? Still, it's the same because you're not gonna. There will be any GLP expressed because of the stop sequence. But when we add Cre, then what happened? Stop's gone. So now then the cell kind of express GLP. The other way around, the opposite way, right? Usually, so all the cells has this transgene in their body, but when only the cells express Cre, become green. So we combine with a specific promoter for this lysam, as we know that it's expressed only in macrophages. So we put this lysam promoter in front of Cre, like we did like K14 Cre, and then combine with this transgenic line, so that this Cre is only expressed where? Macrophages, and the macrophage, only in the macrophage this stop sequence will be deleted so that the GLP can express. So we can label the specific cell type genetically in the mouse. So that's how we see this GLP, the green macrophages. Okay, this is how we make. What about this guy? What do you see? This is on the UV. And this is green fluorescent protein is expressed where? Bone. Yeah. So how would you design this? How would you make this much? Now you know all the information.
With the promoter, it's only in bone cells?
Yeah, promoter. It's a study. Just read some paper about the bone development, bone osteogenesis, and now you know that certain genes are specifically only expressed in bone. So basically here, this is what I'll do. So promoter. Here we use a collagen 1A1 because we know that the collagen 1A1 is only very exclusively expressed in the bone. We take the promoter of core 1A and then just put in front of this GLP, and you have end up this. Again, alkaline phosphatase also is known to be very specific in bone cell or osteocyte. And now you have this. What is it? This is whole body GLP. You can just put the GLP and then put the whatever constitutively active promoter. It expresses everywhere. This is whole body, like whole body knockout, whole body GLP, and this is specific. So basically, you can do whatever you want to do according to your study. So again, now that we go into this, they really want to delete, ablate whole population of not the whole cell, but fibroblast population to get the direct evidence whether fibroblasts promote the tumor progression or suppress. Probably they kind of hypothesized at the time that getting rid of a fibroblast in the pancreatic cancer exert anti-cancer effect. That's right, because that's what all the previous studies showed, that we mixed with fibroblasts and tumor grow better. So now we just try to get it, but those are all the artificial system. We just isolate the cells, grow in the petri dish for a long time, and just mix it and inject it. That's not the typical way or biologically relevant. So we really want to use the transgenic animal system to study this. So they decide to delete or ablate the whole fibroblast population. So this is what they use, kind of complicated, okay? Don't worry. So it's a PTFA. I don't even know what the gene is, doesn't matter. We know that this gene is specifically expressed in pancreatic epithelial cells, okay? We really want a pancreatic cancer model here. And then again, this KRAS, but this is a mutated KRAS. We found this actual same mutation in cancer patient, pancreatic cancer patient. Here, same strategy, we put the stop, LOXP in between. And this is just wild type KRAS. So we just only have one copy of them and modify by genetic engineering. So what happened here without CRE? So this mutate oncogenic KRAS is not going to be expressed. Only this wild type KRAS, which is very important. That's important for normal development because we have to proliferate and do the embryogenesis. So only one copy of KRAS is normally expressed. We have a normal amount of KRAS. So this mice without CRE is totally normal. Okay? Because we want to model cancer progression. We friendly will fine, but over time you drink too much and smoke a lot, then you will get tumor, right? And so we just wait. And the mice were born very nicely, they're totally normal. But when we just add the CRE, then only in pancreatic epithelial cell, okay? Then this pancreatic epithelial cell express now what? Mutant KRAS. So basically it's modeling. So over time we acquire this mutation, right? For many different types of mechanisms, like smoking or whatever. And then so this is kind of modeling. At some point you can start like very early age or very later in old age. You can activate this KRAS, and also they add additional genes, TGF-beta 2. This we know this is a tumor suppressor. Okay, so that we combine this activation of oncogene and then get rid of a TGF-beta because we know that this happens in an actual pancreatic cancer progression. You activate the oncogene and you lose tumor suppressor gene. So we have this, and then you have a very nice tumor progression over time. Okay, kind of basically modeling the tumor progression. So over time they just develop the tumor. We're not injecting any cancer cells. Over time they do. And also they have, because now you have a pancreatic tumor model, transgenic, and what they want is they want to delete fibroblasts. They want to get rid of fibroblasts, okay? So they use the fibroblast specific promoter, this alpha-syndromolactin. It's not important for you, but this gene is expressed almost exclusively in fibroblasts. And then they put this not CRE or JLP is thymidine kinase. Some sort of kinase, okay? Why? I'll explain. So these types of a drug, these types of ganciclovir, okay, it's very, very active. Basically, it's antiviral drug, okay? But when we inject this or we take it, It's not active at all. It's only activated when it's phosphorylated by thymidine kinase. Okay? Only. So it's another control. So only this ganciclovir can kill the cells, and this phosphorylate drug can induce apoptosis to kill the whole cell, right? But you have to have this thymidine kinase. So what if you put this? What's going to happen? You have this mice, you inject this ganciclovir, then only the fibroblasts express TK, thymidine kinase, and the ganciclovir will be activated only in fibroblasts and kill the fibroblasts without harming any other cell type. Although this gene express this ganciclovir, this chemical will be in every cell, but only the cells that express thymidine kinase will be dead. That's how we get rid of this fibroblast. So they did. So they look at the collagen, okay, because the collagen is extracellular matrix being made by fibroblasts or myofibroblasts. So if you fibroblasts remove, then their collagen is gone. So they kind of validate, okay, this system works. So they do look at the survival. What do you see? This is control, okay? This is kind of days, they kind of survive. And when you delete or remove fibroblasts, what do you see? Die earlier than control. So this guy has no fibroblasts because we get rid of fibroblasts, and we got the totally opposite phenotype, totally unexpected. Okay, this is and they look at the number of tumor spares in the spleen. So they look at metastasis as well. If you delete the fibroblasts, you have more metastasis. So the mice died already, and they have more metastasis without fibroblasts. So it seems like fibroblasts is suppressing tumor progression is helping us, right? Suppressing tumor growth is totally opposite from all the previous work that we seen, right? So how would you explain this? Okay, we'll go back to this slide. Where's the fibroblast slide? I think that's how they got the kind of idea. So this is nude mice. What is nude mice? Yeah, of course, there it is. Yeah, it's immunocompromised. Why do we need the immunocompromised mice?
So they won't reject the cells.
Yes, because we want to study human cancer cell line, human cancer cells. So if you put the human cancer in the mouse, then mouse will very nicely and actively and aggressively reject it. Because same way, if you we inject the mouse cancer cell in your body, you're not gonna get any cancer because your immune system like really. So that's what we have to use immunocompromised. So this guy has no T cell, all right? Any idea why we have opposite phenotype? Those transgenic mice, they are immunocompetent. They have no problem in terms of immune system. Right? We do this same, basically similar experiment when the T cell is gone, then tumor can grow really big. But in this immunocompromised, immunocompetent mice, this transgenic line has functional T cells and many other immune system. So I think that's how they got the idea that they look at the immune system. So they look at the T cell, okay? They just compare the T cell activity, or this is the CD4 T cell, which is help kill cancer cells, okay? Like remember the immunotherapy, and this is a Treg cell. So that there is a multiple types of T cells, but T regulatory T cell is actually immunosuppressive. Again, same thing. There is activation of immune response through the CD4, CD8, and then there is a little bit later there's this huge influx of T regulatory T cell going there, suppress the immune system. Because why? Is in the quiz, we don't want overactivated immune system, right? To control checkpoint. As you can see here, if you the fibroblast is gone, you see less cytotoxic T cell, but you have more regulatory T cell. This is a partial mechanism why, if you get rid of the fibroblast, you have less T cell that can tumor cells, and then you have more T cell that can suppress the immune system. So we're talking about the fibroblast tumor interaction. It's not anymore. We are looking at fibroblast and immune system, and then this by through the immune system, when manipulating or manipulation of immune system, that you have a different tumor progression. So basically, what they found is removal of a fibroblast decrease cytotoxic T cell, which can kill cancer cells, and increase regulatory T cell, which suppress other types of immune system, so that the tumor cells will not get inhibited or eradicated by immune system. So that's kind of one mechanism. So that kind of makes sense. All the experiments previously, majority of them use immunocompromised mice that doesn't have T cell, and now they have a lot of tumor growth by adding fibroblasts. But now we use the mice that has intact T cell system. Now we see totally opposite. Think about how easily we kind of misled it, based on the experimental system. All right? And at the time, we really don't care about the immune system very much. Now we know, and this is a T cell activity. So now they have an updated hypothesis based on this. The getting rid of a fibroblast in the pancreatic cancer microenvironment, not anti-inhibit, is accelerated tumor progression through this immune suppression or checkpoint mechanism. So, okay, and then they looked at probably all different types of, but CTLA4, remember, right? One of the gene that identified by James Ellison and MD Anderson got the Nobel Prize. And they did the same experiment, so you just get rid of a fibroblast or control and look at all the lung fibroblasts and immune cells. But now that in addition to get rid of a fibroblast, they add antibody targeting CTLA4, like similar immunotherapy that we use. Okay, it's a checkpoint inhibitor, anti-CTLA4. Now it becomes control. If you add on top of fibroblast gone, then you have a more worse outcome. But if you inhibit the CTLA4, you're back to normal. How would you interpret this data then? So they worked through the CTLA4 mediated T cell suppression. Okay? So if you combine, so this is important because based on all the previous study, we really target this fibroblast. We believe that fibroblast is actually promoting tumor progression. But actually, if you just use only fibroblast targeting, you actually get the opposite result, the tumor grow better. But if you combine with immune checkpoint, like immunotherapy, we might have a better solution, like basically all the combination of all the treatment, because we really have to understand the mechanism, okay? So I want you to think about interpretation. It kind of, it sounds complicated, but not. So just you add the anti-CTLA4, and then now you just reverse or rescue or restore this phenotype, okay? So we're talking about therapy, immunotherapy, molecular therapy, but it's not just therapy, it has a huge influence in the fibroblasts as well, and in microenvironment with the chemotherapy agents. So again, try to understand the transgenic